Hey guys, welcome to Chapter 18, Topic 3. This is the control of cell number and size. So we've talked about how cells divide and we've talked about all the processes and controls they have. But how do cells control, or how does a multicellular organism control cell number and size and make sure that these cells are behaving properly and that we don't end up with tumors and things like that? That's what we're going to talk about here in topic number 3. So here we have our topic outline. We're going to talk about cell number control overview, and then we're going to talk about a process known as apoptosis, and this is programmed cell death. And we're going to talk about how it's regulated, and we're going to talk about signaling involved with it, and then we're going to talk about some of the signals that are used specifically. As always, here's our topic objectives. These are the ones I expect you to have under control before our next exam. So please let me know if you have any questions about these. So how are cell numbers regulated? Organisms rely on regulation of cell numbers in order to live. Otherwise, you're going to end up with all sorts of growths and things like that, or you're going to need to have the opposite effect where cells are not going to be replaced that need to after cells have died. And so cell numbers are controlled by three main things: cell growth, cell division, and cell death. And we've talked about cell growth and cell division already. And cell death is what we're going to talk about now, where we talk about apoptosis and cell survival. So what is apoptosis? And you may have heard it referred to as apoptosis. It's just a regional difference in how you pronounce it. Apoptosis is known as programmed cell death. And this is a very specific, regimented way for the cell to undergo the destruction of itself. It's very different than necrosis, which is where a cell will die due to injury or something like that. And it's utilized, this process is utilized by organisms for a variety of purposes. During development, we all start as this amorphous blob, but what happens is we start removing some cells to help define shape for structures, and that is occurring through apoptosis. We also remove structures that we no longer need. So like if you think about the tadpole, it no longer needs its tail when it starts to become a frog, and this happens through apoptosis. And then the last one's the one that occurs every day in you and me, is balancing out cell division to ensure that we have stable numbers. So as your skin cells are dividing in your lower skin layers, the top ones are dying so that they can be replaced. And so this is it's a constant balance process. So how is apoptosis accomplished? And this is once again through a signal event. Now it's really important to understand that apoptosis is very much an all or nothing event. Once it starts, it's not going to stop. And so it's really, our cells definitely don't want this triggered unless it's time to undergo apoptosis. And so what we see here is we start out with these procaspase molecules, and you can see them here with the different parts to them. And these procaspase molecules have to be cleaved and assembled. And so what you see here is we take off some of the predomains or the protodomains, and then we assemble these large and small subunits into an active caspase molecule. This active caspase molecule is then going to go and create a positive feedback loop that's going to activate many farther down caspase molecules. Caspase is the main molecule that's involved in apoptosis, so it's important that you understand that. And you can see how the different caspases are going to direct different activities involved in apoptosis. So obviously, if this is something that is so all or nothing, and once the triggers are received, how do we stop it from happening? And so what we see here is we see a variety of, or what we see here is the process that undergoes how this is all led to apoptosis. But what we know is that there's a group of proteins known as the BCL2s. These BCL2s will come down here to where you see the apoptotic stimulus and will block that stimulus. And that will stop the release of cytochrome C, and then once cytochrome C is not released from the mitochondria, we will see that the cytochrome C will travel and form this assembly of the apoptosome. And so that's how this all forms into apoptosis. But if the signals are stopped before before cytochrome C is released from the mitochondria, none of this will happen. The cell will not undergo apoptosis. So this whole family is important because the Bachs in the back, the Bach signals that you see here on the mitochondria activating the release of the cytochrome C are also members of the BCL2. So it's two sides of one coin as to whether or not this is activated. So unicellular organisms will use the environment to control cell growth. They will be able to, they're a lot more flexible in that case. Multicellular organisms, your skin doesn't know what the outside environment necessarily is like, and so it relies specifically on signaling from the other cells in the body and nearby cells. And so this is where all that intercellular signaling comes along that we were talking about. And there's three main signal classes. There's the survival factors, the mitogens, and the growth factors. And we're going to talk about each of these signals here in the next couple slides. So the first is survival factors, and this is where we see that BCL2 come into play. Every cell has to receive a signal factor in order to prevent apoptosis. And this makes sense, right? If the body only releases a certain amount of survival factors, then only a certain number of cells are going to be able to survive. And this is an easy way to regulate the amount of cells that we have. So the survival factor is received, it's active, the signal transduction is passed across the membrane, the activated transcription regulator goes and binds the BCL2 gene, makes BCL2, apoptosis is blocked. If this wouldn't happen, then we have that Bax in the Bach that will then cause apoptosis to undergo. And so cells have to receive this signal in order for it to continue to survive and not undergo apoptosis. The next signal we're going to talk about are mitogens. Mitogens are responsible for cell division. And so what they do is they bind, and they will bind to their receptor. The signal will be passed along signal transduction will occur across the membrane and will cause intracellular pathway signaling that will then stimulate the production of all the regulations that are needed to proliferate. One of the key examples of this is platelet-derived growth factor, or PDGF. And what happens is in a blood clot area, the platelets are going to release this factor to the cells around it. It says, okay, folks, we need to make more skin cells now because we need to repair this damage. And so this is how the mitogens work. The last signal is growth factors, and growth factors can be mitogens, mitogens can be growth factors, but they are also two different things. This is where we just stimulate the cell to produce more cellular products. It's not necessarily saying it's going to divide, we just want more cellular products. And so that's what we're going to see here with growth factors. So the growth factor will bind, we're going to pass the signal across the membrane, follow through the intercellular signaling pathways. We're going to increase protein synthesis, but at the same time we're going to stop the cell from degrading cell products. So this cell will get bigger and have more components because we're not breaking things down as fast. We're not breaking things down, and we're making more of them. So it's like a warehouse. So this is the end of topic 3. I know that this stuff was all new in topic 3, so I fully expect there to be questions. So please let me know if you're feeling a little hazy about apoptosis or these signals that help control the cell cycle. And if you have any questions, just let me know, and I'll see you in class.